Vanakkam. Welcome to this video on classics in neuroscience. We have been looking at chemical signaling molecules, that is the first neurotransmitters in the brain. In this video, we will look at catecholamine neurotransmitters, particular dopamine and the topic of monoamine histofluorescence. A big family of neurotransmitters were derived from dietary tyrosine that contained an aromatic ring and an amine side chain neurotransmitters. These were essentially through tyrosine that then becomes dihydroxyphenylalanine or DOPA, which then becomes dopamine, then norepinephrine, then epinephrine. It was known that epinephrine was a excitatory neurotransmitter. At that time, it was kind of understood that epinephrine was perhaps an excitatory neurotransmitter in the peripheral nervous system. It was assumed until the 1950s that dopamine and norepinephrine were practically inactive in the brain until the 1950s. This research was first spearheaded by the work of Arvid Carlsson from Sweden, who was the scientist who first discovered dopamine or provided first evidence for dopamine. He was a Nobel laureate, check Arvid Carlsson and his Wikipedia page or his Nobel Prize web page. So, he found that reserpine and its behavioral effects were essentially due to catecholamine depletion. They found that dopamine, noradrenaline and serotonin were active components found in the brain after reserpine administration. So, they suggested that perhaps that these are neurotransmitters in the brain. It was later realized that depletion of dopamine in the brain, specifically in the basal ganglia, is what causes the Parkinson's disease. It was also found that the debilitating symptoms of Parkinson's disease can be relieved by giving the chemical L-DOPA, L-dihydroxyphenylalanine, which is a precursor to dopamine, which was later converted into dopamine inside the brain, because dopamine as a big molecule cannot cross the blood brain barrier, but L-DOPA can cross. The detailed physiology of this I will present in a different video. I will share with you my video from a different neurophysiology course. You can go through that in detail. The physiology of dopamine and how it causes Parkinson's disease, or the lack of dopamine causes Parkinson's disease, and how L-DOPA is used to treat Parkinson's disease, and what are some animal models of Parkinsonism. All these details I will share with you the set of videos for that purpose. Around this time, a similar set of experiments were performed and evidence to suggest that dopamine was indeed an important neurotransmitter, that the lack of which caused extrapyramidal movement disorders such as Parkinsonism was provided by Ole Hornykiewicz from Vienna. Also Google about him. Okay. So, this set of evidence that was provided by Arvid Carlsson and Ole Hornykiewicz were met with substantial amount of criticism, as in the case of GABA. Why? Because the classic criteria for neurotransmitter identification had not been met, similar to the case of GABA, and so it was not clear whether dopamine was indeed a neurotransmitter. Right. In fact, what was mentioned in the criticism in the 1960s was that there was absolutely no evidence, I am reading from the textbook, there was absolutely no evidence that the catecholamines in the brain act as synaptic transmitters, and any of the theories on a relation between catecholamines or serotonin and behavior is a construction which someday will be amended. This is what was suggested in criticism of dopamine and catecholamines in general. So, it took well into 1970s for dopamine to be established as a neurotransmitter in the central nervous system. So, a major set of evidence supporting dopamine as a neurotransmitter came from the work of Julius Axelrod. Julius Axelrod was invited by Ed Evarts to NIMH. What is NIMH? National Institute of Mental Health, National Institute for Mental Health, rather, for mental health, to set up the pharmacology lab, or to study pharmacology and this line of research. Ed Evarts invited Julius Axelrod to set up a lab. At that time, the leadership of this institute, National Institute of Mental Health, National Institute for Mental Health, was with a person named Seymour Cate. Julius Axelrod mentioned how the research environment was in NIMH, because it is critical that this research environment was what was crucial in promoting the results and the research that was performed by Julius Axelrod and all the colleagues in that institute. I am going to read from the textbook. The philosophy of Seymour Cate was to allow investigators working in the laboratories of NIMH to do their research on whatever was potentially productive and important. Cate believed that without sufficient basic knowledge about the processes, doing targeted research on mental illness would be a waste of time and money. This is what Seymour Cate believed. So, he essentially provided unlimited freedom as long as the problems that you are working on was important and crucial, even if they were basic sciences. In this environment, Julius Axelrod essentially built an excellent and very productive research group, and he mentions about the need for elegance and simplicity in experimental approaches, and in general in research approach. He mentions that one of the most important qualities of doing research, I found, was to ask the right question at the right time. I learned that it takes the same effort to work on an important question as on a trivial one, and when opportunities arose, I made the right choice. This was the statement made by Julius Axelrod. Through all this work, especially analysis of monoamine degradation pathways by Julius Axelrod and his groups, only in the 1970s or in the 1980s, dopamine came to be accepted as a neurotransmitter. We move on to the next topic on monoamine histofluorescence. Evidence to support the biochemical nature or the presence of the catecholamines was established through biochemistry, but there was a need to show this at the neuronal cellular level, and this required development of crucial methodology to do that. The first step in this direction was made by Eino Eranko. Eino Eranko found that cells containing monoamines were rendered fluorescent when exposed to formaldehyde. This was an interesting observation. So, you can identify whether monoamines were present inside the cell by identifying whether there were fluorescence. So, he reported that in frozen sections of brain tissue containing monoamines, he found yellow-green fluorescence when there was exposure to formaldehyde. Right. but the amount of fluorescence was so low, there was low sensitivity. So, there was a need to increase this sensitivity. This was suggested by a person called Nils Ek Hillarp in Sweden, who made some gaseous reagents on freeze dried tissues, brain tissues. Essentially, this gas was formaldehyde vapor, which was found to be an effective reagent that improved the fluorescence, that improved the sensitivity of the fluorescence. So, this was the first method to show the fluorescence due to 5-HT, noradrenaline and dopamine. This was immediately confirmed in vertebrate brain and invertebrate ganglia both. Right. So, this led to a new field of transmitter defined central system, where this monoamine containing neurons were having these neurotransmitters. So, these transmitter defined central systems were identified. So, noradrenergic neurons were found in the locus ceruleus. Dopaminergic neurons were found in substantia nigra pars compacta. Substantia nigra pars compacta and the ventral tegmental area. And 5-HT serotonergic neurons were found in the RAF nucleus. In addition to this, there was this acetylcholine or cholinergic forebrain system was found. So, if you stain one molecule, it revealed entire system. So, this revealed high concentrations of dopamine in the brain, and what caused Parkinson's disease and how Parkinson's symptoms, debilitating symptoms were revived or were managed through L-DOPA. So, this was and the evidence by Arvid Carlson and colleagues was the first evidence to suggest a molecular basis for neurodegenerative diseases such as Parkinson's disease. With this, we come to the end of this video. In this video, we looked at catecholamine transmitters and the topic of monoamine histofluorescence. Thank you very much for your attention.